Hi guys, welcome to our discussion. Today we're going to look at one of the concepts here under genetics, and that is the concept of DNA and RNA. Before we proceed, I'd like to request our members to go to YouTube, search on our channel and try to subscribe. Engage us on the comment section so that we can be able to have a healthy discussion on these different topics that we are discussing. Now, the concept of RNA and DNA, of course, we had mentioned them under the introduction to genetics, and we said that genetics is the study of inheritance and variations. Now, in trying to look for the particular aspect of this topic of genetics, whereby the concept of inheritance and the variation can be displayed, these two specific protein molecules are going to assist us in trying to define a clear difference between these hereditary materials that we refer to as RNA and DNA. Now, in our introduction, we had said that this concept of RNA and DNA, this specifically refers to the protein molecules which are found on the cell organelle that is found inside the nucleus, that is referred to as the chromosomes. And as we said, this DNA or the protein molecules are found alongside this particular part of the chromosome that we refer to as the chromatid. And therefore they appear in form of beads, and they are referred to as genes. So basically, these genes can be referred to as the two molecules of DNA and RNA. And the different names, as we have mentioned, we're going to also discuss about their specific roles, and that's going to give us a clear difference between the two specific molecules. Now, on the concept of DNA, we're going to see that the DNA molecule refers to, this is just a compound name, DNA, referring to deoxyribonucleic acid. And from our previous discussion, we had decided that this basically, the concept of deoxyribonucleic acid implies a ribose sugar, which basically can be presented, or rather, just draw a rough molecule of that ribose sugar. And we see that in this ribose sugar, there's a difference between the ribose and deoxyribose sugar, in that this ribose sugar, this is the deoxyribose, I've basically assumed the other attachments for these specific molecules, specifically for the other parts, because they are just the same, only that the difference appears in this specific part. So at this particular number of carbon, we're going to see that there's an attachment of a carbon at this specific point, where one has got a hydroxide group, the other one lacks a hydroxide group. So the one that has got other hydroxide group at this specific number of the carbon atom is referred to as ribose, while the one that is lacking is referred to as deoxyribose. So therefore, these concepts come from this specific point, and we said that the ribose molecule is a sugar of the pentose class, which basically appears, or maybe is dominantly can be able to be seen appearing in nature. So we're going to also look at the difference between the deoxyribonucleic acid, now that I've explained what this compound specific name refers to, insofar as the deoxyribose is concerned. Now, for nucleic acid, we're going to see that these are specific bases. These are specific, is determined by the specific bases that we're going to see, which are going to be attached in these two specific molecules. Now, for RNA, this is the ribonucleic acid, and I've shown the difference between the concept of a deoxyribo and ribonucleic acid. So this one defines the difference between these two specific molecules. Now, for DNA, it's made up of a double helix strand, which basically appears in this form. It's the first strand and the second one is going to be appearing in this form, so it's going to form what we call a double helix. This is a strand, double helix, a strand, and this strand twists and intertwines each other to form this specific double helix. Now, along this strand, we're going to see that specific molecules are going to be attached, whereby each strand of this DNA is going to contain a sugar and a phosphate molecules, which are going to alternate in this specific order. But in trying to, I'm going to give an illustration which is going to give a clear picture on how these sugars, of course, interchange or maybe alternate. Now, to each sugar, bases are attached using a particular set of arrangement. And as I've said, I'm going to illustrate that. Now, there's a difference between nucleosides and nucleotides, and whereby we're going to see that these specific molecules are going to fall under this specific category, whereby they are going to contain, we say that for nucleosides, they contain the sugar and a nucleic base or a nitrogenous base, whereas for nucleotides, they contain a sugar and then they contain a phosphate molecule, and then they contain what we call a nitrogenous base. Therefore, this one has 3 constituents while this one has 2, and therefore we're going to look at this specific case whereby these 2 strands of molecules are going to fall under the category of nucleotides. And therefore, these specific bases are going to be joined together with these particular sugars and phosphate molecules in a specific order that I'm going to mention shortly. Now, the bases that I've mentioned, which are contained in this specific DNA and RNA, are given as thiamine. We have uracil, and we have adenine, we have guanine, and cytosine. So these are the nucleic bases that we're going to see being displayed in this DNA and RNA structure. But basically, for the case of uracil, we're going to see that in the absence of one of the bases, it's going to be replaced by another one. So basically, at that specific point, I'll mention the difference between the two. But we're going to see that these specific bases have what we call the attachment, or the particular set in which they are going to alternate, whereby we're going to see that these attachments can be referred to as receptors. Now, in biology, we're going to look at the different ways in which we can refer to as receptors. But basically, I can just do a geometrical representation of what I can refer to as a receptor. If I have a molecule of this nature, and there's another one, a corresponding molecule of this nature, and I refer to this one as molecule A and B, and I'm trying to bond these 2 molecules, I'm going to see that this molecule is easily going to fit into this particular set of the second molecule at this specific point, respectively. Therefore, I can refer to molecule B as a receptor molecule. Now, suppose I have a different molecule which I'm going to illustrate, which has the same feature as molecule A, but I have a different molecule B which has that specific point. Now, this molecule A and B are not going to join together, because this point of this meeting point, or the point of reception, is crooked in a way that it cannot fit in this particular molecule. So therefore, molecules which are able to accept one another as, or maybe conform and maybe join together harmoniously, like the one I've demonstrated, therefore this can be referred to as a receptor molecule. Now for these different bases, we're going to have specific initials which we are going to refer to them. For instance, for thiamine, it's going to be given an initials T, capital T, uracil, capital U, adenine, capital A, guanine, capital G, cytosine, capital C. And now in using these initials, I can be able to write this as T and maybe A, respectively. So therefore, respective bases are going to have their receptor, their individual receptor bases, which they are going to have a reception for. And this is going to be a specific kind of bonding. Therefore, the arrangement, the double helix arrangement of this DNA structure is going to have a particular base corresponding to the opposite one, which it can be able to do a reception for. So that is going to be clear when we're going to do an illustration for that specific case. So basically, to make a structural difference between the two insofar as the rearrangement of those bases are concerned, I'm going to give a clear cut between the two nucleic acids that I've listed on the board. Now for DNA, it's going to contain these specific molecules which are basically thiamine, adenine, cytosine, and the guanine. Now for RNA, the thiamine is going to be replaced by another base which is going to be known as uracil. So therefore we're going to have the uracil and then adenine and then cytosine and then guanine. So basically giving the initials, then therefore for the two strands for for the two molecules for DNA and RNA, therefore DNA is going to contain thiamine, adenine, cytosine, and the guanine, and this is going to contain uracil, adenine, cytosine, and the guanine, and they're going to bond according to the receptor molecule demonstration that I'd given above. Now I'm going to basically try to illustrate this, but we're going to see that in its formation for DNA and RNA, this one has a double helix strand, while this one has got just a single strand that is maybe twisting in that specific order. Now I'm going to give an illustration of how this particular respective bases are going to bond. Now we're going to see that maybe thiamine has bonding with a respective base, which has a receptor site for it, and basically this is going to be the case for the other molecule. Now the illustration that I've given on the board is showing a respective bonding for these specific bases, whereby we're going to have this particular DNA or RNA molecular strand being shown by this straight set of arrangement, and this one, but as we said, this one is a double helix twisted, whereas this one is just a single strand that formed as that specific formation. So we're going to have this one bond, adenine bond to thiamine, guanine bond to cytosine, thiamine bond to adenine, just as in the first case, etc. But for the RNA strand, we're going to have a uracil now that it's missing at this specific point. So in any case, if we have this specific strand that is representing RNA, then therefore we're going to have a replacement for thiamine being replaced by uracil. So that's cell specific, and for S and P, this one refers to sugar. This is a sugar molecule that is at this specific point, and then P refers to a phosphate molecule, which we are going to see shortly. Now the above illustration is basically going to give us a summary of what we have discussed concerning the structure of the DNA and RNA, whereby we have said that for DNA it consists of 3 parts, which is a phosphate molecule joined to a ribose sugar. Now that we have expanded on what the ribose sugar, deoxyribose sugar means, it's a class of a pentose sugar that we said which is naturally found and can be represented as such. Now this is a base whereby we're going to look at different bases in nucleic acid that we mentioned. And now we have different types of bases which I've already demonstrated using the two strands, which is adenine, guanine, cytosine, and the uracil. And now in this case, for the DNA structure, it's going to have thiamine, while for the RNA nucleotide is going to have uracil in place of thiamine. Now I've already said that for the DNA structure, it's made up of double helix strands which are intertwined as such, while the RNA is made up of a single strand that consists of these different bases and attachment shown above. So as I've said that the basic building block or the rearrangement of these specific strands for DNA and RNA is made up of an alternating phosphate and sugar molecule which is attached to a specific base, which is bonded to a base of another strand. So this specific alternation is what forms basically this what we call the double bond, and they are linked together by a covalent bond that is known as a phosphodiester linkage, which we are going to basically see in this specific illustration when we're going to give a full strand of the representation for this DNA and RNA respectively. So basically in this demonstration, the phosphate is going to be represented by this particular P for phosphorus, which has an attachment for oxygens at this specific point. And at this number 4 carbon, when we are trying to name this specific sugar, this is 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So basically we're going to see that for 5 and 3 prime, these are the attachment areas for phosphodiester bond that we have mentioned. And this is the next kind of bonding is going to take place at this O group of the OH molecule that we have said. And we talked about the concept of a deoxyribonucleic acid, whereby we say that for ribose sugar it lacks the OH group, but for the dioxyribose sugar, which is contained in this case, is going to have a OH at this specific point. This is for the 2 prime. Now we're going to see this difference being manifested, of course, in this specific area, whereby the difference between the first and the second molecules, that the 2 OH group here and this one. Now for the ribose sugar, this specific molecule contains H, while for the second one it only contains a OH group. Now this is the arrangement that is going to resonate for these two specific set of structures for DNA and RNA respectively. So in our next discussion, we're going to see an expanded form of this specific structure for RNA and the DNA, so that we can have a clear understanding. Or basically I can just have an illustration for this specific kind of structure, so that we can be able to have a deeper understanding. Now basically we're going to have that one in our next video, since it's a discussion that is somehow expanded, so that we can have a clear cut between these two particular illustrations for the concept of the molecule of RNA and DNA respectively. So let's catch in our next video, which we're going to show the difference between bases in nucleic acid so far as their structures are concerned, and perhaps the linkage that is going to happen between these double helix concepts that we have talked about, which basically it can be referred to as the attachment between a base and a base, which we are going to refer to as hydrogen bonding. So that is it for now, guys. Bye. Let's meet in our next video.